right. Where we left off, we've been talking about models of speciation, how new species come into existence. And in my evolution course, this takes a couple of weeks. But the simplest model of speciation is speciation called allopatric speciation, which applies to starfish. Right? You've heard of allopatric star? Like if Patrick Starr's species got divided in two, you'd have two species, and that would be allopatric star species. Okay, sorry, not working. Where a population gets divided into two by a barrier that the population can't ordinarily cross. And once you have two separate populations that cannot usually exchange members, several different processes will make them become increasingly genetically different from each other. And a beautiful example of that was caused by plate tectonics when the Isthmus of Panama formed starting about 4 million years ago, having to do with the big subduction zone in the Pacific that built that isthmus. Species that previously had lived on both sides in what is now both the Caribbean and the North Pacific were divided into separated populations that couldn't cross the isthmus because sea snails can't crawl through rainforest most of the time. And so they've separated into separate species, and we have pairs of species in the Caribbean and the North Pacific that are very similar to each other, like the Caribbean rock snail Purpura patula and the Pacific rock snail Purpura panza, the Caribbean deer cowrie Cypria cervus, and the Pacific deer cowrie Cypria cervinetta, and so on. We can do a little bit better than this, but I've got to introduce one more topic. Darwin had realized that if evolution was true, it gave you a new way to classify living things. You can classify them based, as best you could, on common ancestry, not simply what they look like, because that's not foolproof, but on what their closest relatives are. I think the example that I gave in lecture was, if you look at the phone directory, you remember what that was, for Conway, we're practically crawling with Freyaldenhovens. And the simplest explanation for that would be that all of the Freyaldenhovens are descended from some guy named Mr. Freyaldenhoven, who came to Conway and had kids, and they had kids, and they had kids. If anybody is curious, Walkertown, North Carolina, outside of Winston-Salem, has an unusually high concentration of Wagoners. That happens to be where my father's family is ultimately from. And for Conway, you could do the same thing with the Hambeacons and the Siebenmorgens and the Otts and the Maltbias. There's a number of families with pretty unique names that are found here, and the simplest reason for why is common ancestry. And if you could work out what that pattern of common ancestry would look like through time, it would look like a great tree with the roots down around 4 billion years ago when the very first life form coalesced out of the primordial slime. How that happens is not something I care to get into. That could be a whole course in itself. We don't know everything, but we've got some very nifty leads. But whatever the earliest life form was, that gave rise to new species, and those to new, and those to even newer. And what you get, rooted in deep time and spread out through the entire Earth would look like a tree, if you could see it all, with closely related species on close branches of the tree, more distant relatives on more distant branches. The fossils are, the dead and broken branches that fill the crust of the Earth, like the dead and broken branches that filled my driveway after the hurricane last week. Fortunately, not filling my life.
Hey, Dr.
Yes?
Wagner.
Yes, who said that?
Our slides online are looking a little bit weird.
Okay. Well, maybe that's just a funny.
They're like zoomed in a
Okay,
lot,
hang on.
so we can't see the
Okay.
whole slide.
Here, I'll do what. Okay, I'll do what always works. I'll stop presenting and then start it again. Never fails, does it? Okay, is okay, is that any better? Is that
Yes.
better or is that the same?
It's better.
It's better.
Okay, I did nothing but stop presenting and start it again, but evidently that works. You heard the one about the 3 engineers in a car that's driving through the desert. All of a sudden the car starts making a horrible noise and steam starts shooting out from the hood, and before you know it, it has crapped out dead on the side of the road in the middle of the Mojave Desert. The mechanical engineer says, clearly we've blown the transmission, I'll get out my tools and rebuild the gearbox. The electrical engineer says, no, clearly we've blown the wiring system, I'll get out my pliers and I'll have this fixed. The computer engineer says, why don't we just close all the windows? Thank you very much. That raises the question, this whole thing about the tree of life, about how can you reconstruct the tree? How do we know what that looks like? Skipping over an awful lot of details, we'll jump up to the 1950s to a guy by the name of Willy Hennig, a German entomologist, no less, who'd come up with a method for trying to be rigorous about reconstructing what the tree of life looks like. A method that he called cladistics. All right, I always seem to say it wrong. I'll never test you as to what Greek and Latin roots mean, but klados happens to be Greek for a branch. So cladistics is just branchistics. And it goes like this. Evolutionary changes are relatively rare. Most of the time, if you have, how does the lyrics go from the musical The Fantastics? Plant a radish, get a radish, never any doubt. That's why I like vegetables, you know what you're about. Plant a cabbage, get a cabbage, not a sauerkraut. That's why I like vegetables, you know what you're about. Most of the changes that we see, most of the time when you plant one type of living plant, you get the same thing back. Mutations do happen, selections do happen, but it's not as if you could plant a radish and 3 generations later you're pulling up sunflowers or something like that. You don't get that kind of radical change happening rapidly all over the map. Most of the time, heredity is pretty conservative. Offspring look an awful lot like the parents. Occasionally, you'll get what we call convergent evolution, where 2 or more lineages that are not particularly related to each other develop the same trait or the same feature. probably because they are subjected to the same environments and the same selective pressures. I don't have pictures of this, but in Africa, there are a whole bunch of plants in the family Euphorbiaceae that are succulent and look a hell of a lot like cactus, but in fact they're not. They're not even closely related. If you've ever seen documentaries on Australian wildlife, you might know that Australia has marsupial anteaters called numbats. And marsupial, well, the Tasmanian wolf was a marsupial carnivore until it died out in the, well, was driven to extinction in the 1930s. And it looked very much like a wolf, but it's not closely related at all. There's even a marsupial mole in Australia that, well, is blind. It digs through the soil, not the soul. That'd be scary. A mole in your soul. But anyway, it's not especially closely related. So you can get two different lineages of organisms hitting on the same type of adaptation independently of each other. But it does happen, but that's even more rare. So the idea is that if you have two or more taxa, and in case anybody forgot, a taxon just means a biological grouping, right? A species, genus, order, class, what have you. If two or more share a trait in common, the simplest explanation is that they inherited it from a common ancestor. Think back to our Freyaldenhovens. Last names do change. My great-great-grandfather's great-great-grandfather was a Wagner from Germany and spelled it W-A-G-N-E-R. And here I am, a Wagoner, 2 G's, 1 O, 1 N. So clearly somewhere down the line the spelling shifted a bit. So last names can change, but they don't normally change very often, right? It's not like I'm a Wagoner and my dad was a Wagner and his father was a Schmeckelhofer and his father was a Schmendrickberger or whatever, right? We don't change last names every generation. Same thing with biological features. Those don't change very often. So just as if two people share a name like Freyaldenhoven, the simplest explanation is that they inherited it from our common ancestor, Mr. Freyaldenhoven. Now, maybe not. Maybe there are, maybe two people independently decided, hey, I'm going to change my name to Freyaldenhoven because I like the sound of it. It's possible. But the simplest explanation is that they share common ancestors, right? Same thing in biology. If two or more taxa share a trait in common, the simplest explanation is they got it from a common ancestor. And the more traits they share, the likelier it is that they inherited it from a common ancestor. So there's some convergence for you. On the left, that is a flying squirrel of the sorts that I used to have in my old neighborhood. I remember walking to school one day and one of them just kind of glided right past my nose. That was neat to see. So there they are. There's one on the left. On the right, that's an Australian mammal that also has a gliding membrane connecting its wrists and its ankles. This one's called a sugar glider. And they both climb up trees and then can glide down. They fly, body shape is very much the same. Flight capabilities are very much the same. That gliding membrane actually has a name. It's called the patagium. But if you look at all of their features, each one shares far more in common with very different groups of animals. The flying squirrel shares a lot more features in common with rodents, like, for example, incisor teeth that grow throughout life. The sugar glider shares a lot more feature with possums and kangaroos and things like that, like giving birth to underdeveloped young and letting them mature in a pouch, right? The same way kangaroos and possums do. There's other features of the skull, the teeth, the pelvis, all kinds of anatomical features that the flying squirrel shares with rodents. And there's features that the sugar glider shares with marsupials that they don't share in common with each other. So the simplest explanation is the patagium is convergent evolution because it's the only feature that these two critters have in common. In every other respect, each one is more like a different group than they are like each other. Okay, this being marine biology, a very good marine example of convergent evolution would be whales and fish. Up there on the top, that's a bluefin tuna. Down there on the bottom, of course, that's a dolphin. And they've both got highly streamlined bodies. Next week, we'll actually look a little bit at engineering and look at how what sort of bodies are most efficient at swimming with a minimum amount of drag. And there's really only one way to make a low drag swimming body. And both tunas and dolphins have it. So they're both highly adapted towards long distance, high speed swimming, which they can do very efficiently. They've both got movable fins both for propulsion and for stability. But whales have, oh, good grief, mammal-type skulls, a diaphragm, pelvic bones, hand bones in the fin, teeth in sockets, warm-bloodedness, although actually tuna have that too, now that I think about it. The blowhole, that's the nostrils that have moved up between the eyes. No nuclei in their red blood cells. Whales have an enormous number of features in common with other mammals. Milk, feeding their babies with milk. Tuna have many more features in common with other bony fish. Fins supported with rays, hypermobile skulls with independently mobile upper and lower jaws, gills, egg laying. Tunas have much more in common with other fish than they have with whales when you look at the body as a whole. So the similarity here is convergent evolution. The majority of features, if you take all of them, would group dolphins with other mammals and tuna with other fish. Although there was a famous Monty Python skit where there's a guy sitting on a park bench. Someone walks up and says, "You know, the whale's not really a fish." I says, "It's not?" "No, it's an insect." And that's it. Monty Python, what can I say? Okay, so if you try to draw up a tree of life for n species, the total number of trees that you could draw is 2n minus 3 double factorial. What the hell is double factorial? Factorial is a number times the number minus 1 times the number minus 2, ... all the way down to 1. So 10 factorial is 10 times 9 times 8 times 7 times 6 times 5 times 4 times 3 times 2 times 1. Double factorial, you go every other number. So 10 double factorial will be 10 times 8 times 6 times 4 times 2. If we had 10 species, and we wanted to draw up a tree of how they're related, the total number of possible trees turns out to be 17 double factorial. 17 times 15 times 13 times 11 times 9 times 7 times 5 times 3 times 1. And that's something on the order of 20 million. If we want to do even more species than that, we rapidly exhaust the capacity of computers to study them all. Okay, 34 million, not 20 million. I should be reading my own slides. How do we pick the one that's best? How do you find the needle in that enormous haystack? The best tree is the one with the maximum number of shared characters and the fewest number of convergences. Don't worry about the god-awful synapomorphies and homoplasies terms right there. The tree that gives you the maximum number of shared characters is the one that you go with. A tree that puts dolphins with other mammals and tunas with other fish is to be preferred to one that puts dolphins and tunas together. It's basically a matter of picking the simplest tree, the one that makes you make the fewest assumptions as to how evolution has gone. And we call that the principle of parsimony. Okay, Wolfda, I just realized something. In the syllabus, I've got these exercises that I want you to do. If we were meeting in person, we do them in the lab for about an hour. But under the circumstances, I'm trying to move them to the virtual sphere. I won't make it ruin your Labor Day vacation, but after Labor Day, I'm going to have you guys do a little hands-on cladistics investigation that hopefully will make these concepts a little more clear than me just standing in here and yelling them at you. Okay. If you have, you know what, I'm going to keep going. Okay, here's some critters. The way we used to classify these arthropods, these invertebrates that have jointed legs, is insects like the butterfly and the beetle on the right, together with some wingless relatives of insects like springtails and things like that, used to be placed in a group called the hexapoda. Hexapoda, 6 legs. All the critters on the left classified in the group Crustacea. That, of course, includes crabs, shrimp, and lobsters and things like that. It includes brine shrimp and copepods. You'll be seeing copepods again because they're very common in the oceans. And then, of course, crustaceans in the oceans, many of them are quite delicious. Crabs and lobsters and all of that. And there's a parasite called a fish louse that's a funny-looking crustacean. And all of these have been placed in their own group, the Crustacea. And we used to consider these separate from each other. I mean, related ultimately through common ancestry, but different. But what we've been able to do is take DNA from these critters and work out the order of base pairs. You know, read the ATG, ACC, GG, ATT, whatever. Compare DNA from all of these organisms with all of the others. And it now looks as though this old group that we used to call Crustacea also has to include the insects as well. There's a branch point down at the down below, right there. That point where the branches are coming from represents a common ancestor of all of the critters that you see. That common ancestor gave rise to all of the Crustacea, but it also gave rise to all of the insects as well. The last common ancestor of everything we've been calling a crustacean was also a common ancestor of everything that we've been calling an insect. And so now we don't separate the two from each other. All of the critters that you see are part of a large group called the Mandibulata. So we don't split them now because we're trying to make our classification scheme follow common ancestry. Okay, I started with the Isthmus of Panama. Let's get back to it. There was a study about 10 years ago on Pacific and Caribbean species of crustaceans in the genus Alpheus, known as the pistol shrimp. Pistol shrimp are incredibly cool. Look at that big claw. What they're able to do with it is bring up the claw and snap it in such a way that they actually very, very briefly create a bubble of vacuum in the water. The vacuum bubble immediately slams shut, and what that creates is a powerful shock wave, which means that the shrimp can walk up to something it wants to eat or a predator that might eat it and hold up that claw and go bang and stun its prey or stun the predator and either get away or go up and eat the prey. Hence the name pistol shrimp. This thing will walk up and just go bang and create a disabling shock wave. Kind of a cool trick. Good to know that the ocean, they have the right to bear claws. Get it? Not just bare arms, but bare claws. Okay, sorry. Now, okay, this is more than 20 years ago, 1998. They did a study of DNA from species of Alpheus, the snapping shrimp, and they took them from both sides of the Isthmus of Panama. And we'll put the colors in. I've put in green each one of the Pacific species, and in blue, each one of those is a Caribbean species. You don't even have to read the names. But if you look up at the top, you can see there's a Caribbean species called Estuarensis, and its closest relative is a Pacific species called Columbiensis. And then below that, there's a Pacific species called Antipenultimus, whose closest relative is a Caribbean species. And you can see this pattern, and you're only seeing a part of the results. They found about 20 pairs of species where the closest relative of the Caribbean one was the Pacific one. And the simplest explanation is there used to be 20 ancestral species of snapping shrimp. And then the isthmus formed, and each of those 20 species of snapping shrimp got split into two, into Caribbean and Pacific populations that were no longer able to mingle. Were no longer able to breed with each other because there's a big bunch of land in the way. And so over the past few million years, the separated populations of Caribbean and Pacific shrimp have diversified into separate species, but they're still each other's closest relatives. Each Caribbean species' closest relative is a Pacific species, and vice versa. So that's the kind of insights you can get into the processes that have shaped the oceans and the life in them when you put together some plate tectonics and you put together some evolution. And we'll be seeing more examples of this as time goes by. Okay, well, we started a little late, and I think we might actually finish just a little bit early. We'll go ahead and stop the recording. Have a good Labor Day. Be safe. Don't get infected or the governor's going to get mad.